Hi folks, welcome to our series in histology made simple with drawings and diagrams. As you can see, we only have here a whiteboard and a few markers, which is what I would need if you would. I would have to individually explain you these facts. I hope anyway that they are useful. As you can see, to draw a cell, you only need to do one circle and another one, and perhaps for the nucleolus you need another one. Well, it's easy, right? And most of you will probably say, oh my gosh, this cell, I've had so many times to read and to encounter the cell in high school, middle school, and even college that probably I know everything. Perhaps they do, perhaps you do, but perhaps there are a few facts that you need to a little bit, clarify. Before we, start with the cell, let's say a few facts about the cell, just to give it a little bit of perspective. For example, I'm going to ask you some questions. I know that you cannot answer me, but I'm going to imagine that you do. For example, do we share the same cells with a dog, a mouse, or a flea, for example, or any animal, for example? Yes, they are exceedingly similar to ours. Not in the grade of complexity, but many of the organelles are there. How about the plants? Lettuce, tomatoes, what have you. Yes, the cells of the plants are strikingly similar to ours, except for cell walls or some other organelles. So it's amazing, right? As far as, for example, can you see the cells with your naked eyes? You would say, no, it's impossible. That's why we need a microscope. You will be right, but you would be also wrong. Because there's one cell that you can see with your naked eye. Which one it is? The ovocyte, the feminine cell, the ovocyte. It is about 1/10 of a millimeter, so it's a speck of a whitish thing that you can see with your naked eye. What about the sperm head? The sperm head is one of the smallest, if not the smallest cells. It's about 7 microns compared to 100 microns of the ovocyte. Now the other question is, what about the size of the cells? Is a cell of a mouse smaller than that of a giraffe or an elephant or a whale? Do you have different sizes according to the size of the animal? The answer is no. All living creatures essentially share the same size of cell, about 20 to 30 microns average. So it's amazing that a whale may have so many cells. For example, and how about us? How many cells do we have? Did you count them this morning? No, I hope so. Probably not. You probably not, because some people estimate that we have 40 trillion cells. If you remember that counting 1 trillion, one number a second, it would take about over 31,000 years. So our number of cells is huge. Finally, I will mention something. Some organisms are multicellular, like us. Some others are unicellular, like an amoeba and other organisms. Now, are we really multicellular, or are we really unicellular? Of course, you said we are multicellular. You just said we have so many cells. But all our cells are exact copies of the first cell that was formed at conception. That cell only just divided into, and etc., 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 trillion times. So actually, all our cells share an individual makeup, are just totally different. And now, essentially, how do we know that? How do we know that our cells have the same DNA? Do you know that? How about the cloning experiments? Clonation takes one cell, any cell essentially, or whatever fits better, takes the nucleus apart and puts it into an empty egg and hopes it with a lot of effort to start reproducing. And what does it do? It reproduces exactly the same animal. And they have been trying to do it with humans. I don't believe that has been feasible so far, but that is what they have been trying. Let me get a glass of water. Now, let's start with the cell. And before starting with the cell, I should have to mention something. If somebody asks you, what is your body made of? And you said, cells. That would be a correct answer, but you would have to be forgetting something very important, which is the extracellular matrix. In other words, around our cells, we have fibrous tissue, bone, cartilage, which form ligaments, capsules, etc., etc. Your bones themselves are made, no, they are not made of cells on top of the other. They are made by cells. So the little tiny architect or engineer, which is a cell, makes a whole body made of cells and another set of elements such as bones, cartilage, and other which form part of the extracellular matrix. Okay, let me erase this right now. If we go to the cell, let's start with the first part, which is the membrane. I drew it beforehand, so it's because of that, it's not easy to make it around like that as it seems. Now, the membrane is, for many years, was considered just like a little membrane, something very simple, just like the rubber of a balloon. But you know that it's not like that. It's not completely like that. It's actually not one layer, but a bilayer of phospholipids. And those phospholipids are essentially, I drew it for you, and are made of a nitrogenous compound, which may be choline, serine, or ethanolamine, a phosphate bridge, and glycerol. What is attached to the glycerol? Fatty acids. What kind of fatty acids? No, saturated and unsaturated. Those that are unsaturated are a little bit kinked in the mid portion. So essentially, that. Now, this was discovered about almost over 50 years ago by Singer and Nicholson, the fluid mosaic model, and pretty much tells you that these structures are very functional. The inner and the outer portion are hydrophilic, so they love water, and the central portion, the area of the fatty acids, is hydrophobic. Now, are we done with the membrane? Is that all we have? If you say not, you probably are right, because we need to add here the proteins. Some proteins enter about in the outer part of the membrane, some others go all the way down, and they have different functions and as receptors, and sometimes in facilitated diffusion and many other functions. So that the membrane is actually studded with multiple bumps, which are those proteins, extremely important. Now, at this point, should we say, are we done? Are we done with the membrane? Those who say no are probably right. We need to add the series of carbohydrates, actually polysaccharides, that are attached to the proteins, especially those that appear on the outside of the membrane, which have a series of mechanisms like self-recognition and et cetera, et cetera. So in other words, the cell is not as smooth as we thought. It's a little bit fuzzy and bumpy. Those components of the cell are in perpetual removal as the rest of the cell. The cell is like a microcosmos that is perpetually changing all the time. Now, let's say something, like a question. How do substances go through the membrane? One of them would be just by concentration, gradient of concentration without any difficulty, which is the passive diffusion. Obviously, that will be oxygen, CO2, and lipids. They just go back and forth without much difficulty in view of the concentration. Some other substances need a protein to help them go in or out, although they respect the concentration. I would say put here a protein, and then we'll have the passage of substances that go in way or one way or the other. And which are these? First of all, of course, glucose and amino acids and water and ions. Finally, there are a couple of ions that need to be pumped in and pumped out and require an active mechanism. Now the question, the million-dollar question is, which are those ions that need to be pumped in or out? Oh, those are potassium and sodium. So we have a sodium and potassium ATPase that involves in this active process of pulling in and out potassium. Now let's pay our attention to the nucleus. That's what we're trying to simplify. First of all, there is something that needs to be changed in this diagram. We have one membrane in the nucleus. Is there any difference? Is the nucleus any different from the remainder of the cell? If you said yes, you're right. You need another membrane. Let me excuse me if I go this way. The nucleus has two membranes. Two complete membranes. Now, is there any difference between the inner membrane and the outer membrane? Raise your hand. No. Okay. You have ribosomes attached to the outer membrane. And so it's kind of reminds you of the rough endoplasmic reticulum, right? Now, I would ask you something. Is there all that we need to know about the outer portion of the nucleus? There's one detail that we need to add, which are the pores, which are essentially openings that allow the substances to go in and out from the nucleus to the cytoplasm. Now, those pores are not just holes the way I just put with my eraser. They are complex structures on the molecular level made of 50 proteins, each one of them. But anyway, they allow RNA and proteins to go in and out and other hormones as well. So now, when we start looking at the nucleus, you will notice that I use the blue marker to mark the nucleus. And that's not just because I like the blue marker. It's because the nuclei appear blue in H&E sections. Why? because hematoxylin is a basic marker and is usually stains negatively charged substances like chromatin. In other words, whenever we have the nucleus stained by hematoxylin, strongly. Now, one little detail before we enter into the nucleus is to know if you have here some ladies will notice that their nuclei sometimes in several cells have a little bump that protrudes slightly outside or more in the neutrophil, which is the bar body. The bar body, let me just write it down. What is the bar body? The bar body is one of the X chromosomes that is inactive in women. Why is it like that? There may be many theories, but who understands women anyway, right? Anyway, they only need one X chromosome. They are strong. Now, if you focus on the chromatin, I highlighted some areas that are dark and some areas that are pretty much clear. Though the areas that are dark have a different name than those clear. The dark areas are called heterochromatin. And the clear areas, euchromatin. Euchromatin. What is the difference? What do they do? We just give them names? Or what is their function? Which ones are active, the euchromatin or the heterochromatin? You know, one would think that's the dark ones, but are the clear areas, the euchromatin, that is active. Why is it active? Because although each cell has all the library of genetic information, it only uses a certain amount of information, and the rest is usually attached to the periphery of the nucleus, or giving the cell some kind of a geographic appearance. Now, when you focus on the nucleus, you heard that you may find an area that stains stronger and called the nucleolus. The nucleolus is sometimes so strikingly clear that you would believe that's an organelle bound by a membrane, and it's like an organelle of the nucleus. However, it is not. What is the nucleolus? Nucleolus is actually an aggregation of genetic material that includes segments of 6 different chromosomes put together in order to function. What does a nucleolus do? It produces ribosomal proteins, and also it assembles ribosomal RNA and ribosomal proteins. I repeat it again. Ribosomal RNA and also assembles ribosomal proteins. Now, if I ask you which cell would need to be have a big nucleolus, and if you see a cell that has a big nucleolus, what does it mean to you? It means that the cell is in protein synthesis, because the RNA and the ribosomes form part of the rough endoplasmic reticulum, and we are dealing with protein synthesis. I'll remind you that cancer cells are very active and sometimes they have huge nuclei. Let's start now saying a few words about the rough endoplasmic reticulum. Rough endoplasmic reticulum. It's difficult to really draw them, but if you make several lines like this, you give the impression that you have a series of flattened tubules and sacs, and structures that are vesicular looking, flattened together. But there is one little extra detail that we need to put here in order to make it a rough endoplasmic reticulum. It has to be rough. Not because somebody touched it, because it kind of looks rough, because it has myriad of ribosomes attached to them. Now, on the other side, if you kind of go to the opposite side of the cell, just for completeness sake, and you draw the smooth endoplasmic reticulum, it does not have ribosomes. Now, I would say something. In order to kind of understand the function of the ribosomes, you would have to go to protein synthesis. And that is not an easy task to do in a few words, but we'll try, okay? The DNA, the information of the DNA needs to be, for example, suppose that this is a chromosome, and obviously it has the same sequence, the sequence that you know, the genetic code in the usual nitrogenous basis, and we have exactly the information to make a protein. So we need a messenger to take this message to the ribosomes to make proteins. So we have a RNA messenger RNA will copy the information and take it to the ribosomes. Now the ribosomes, each ribosome is actually extremely complex. It's, from the molecular point of view, is like a city. But we can actually maybe simplify it. It has about the shape of an 8, the number 8. And the messenger RNA enters in that structure. Now, what do we need afterwards? We're making proteins, so we need amino acids. Where are the amino acids? Let's say the amino acids are in the cytosol, in the cytoplasm. So let's make it an idea that these are the amino acids. They are in a haphazard fashion, and they just need to be arranged in a very orderly fashion. So the transfer RNA, I'll make it blue this time, is going to take one by one and set them exactly in the same order from that of the DNA. And what do we get? Voila, we get on the middle of the, in the center, in the lumen of the rough endoplasmic reticulum, you end up a series, a protein. In other words, a sequence of amino acids that mimic exactly the information of the DNA. Now, through multiple processes in the RNA, in the ribosomal RNA, the protein is going to be modified in its tertiary quaternary structure. Sulfate binds are going to be introduced, but we have to go ahead. Now, let's talk about the next structure that we need in order to keep on our discussion. This is the Golgi apparatus, which has essentially similar structure of that of the raw endoplasmic reticulum, but it's more concave, almost like, and has a trans and a cis phases. And what happens next? Well, the proteins that were present within the rough endoplasmic reticulum are going to be transported to the Golgi. apparatus, and they are going to be glycosylated, they are going to be modified in different ways. And after that, they have different endings, I would say. Excuse me. They can stay in the cytoplasm, or also they can, let me put this thing away. They can actually fuse with the membrane and be secreted and be, for example, part of the extracellular matrix, such, for example, the fibers, fibers produced by the fibroblasts. Some of them will be part of the cell. I forgot to tell you that not only the ribosomes can be associated to the rough endoplasmic reticulum. Some of them are present in the cytoplasm, and they the proteins will remain within the cytoplasm. Okay. Now, on occasions, the cell will phagocytize, for example, a bacterium, like in neutrophils, monocytes, etc. And the bacterium will be inside the cell. And how do we kill the bacterium? You cannot use Lysol or something like that. You need one of these vesicles that include enzymes that will fuse with the phagosome and phagolysosome and destroy the bacterium. Some of the other, some other of these vesicles require special receptors that will be recycled, but we function in a similar fashion. Okay. Now, regarding a few words about the smooth endoplasmic reticulum. Well, they actually, they synthesize lipids and membranes, and they are particularly useful in the hepatocyte, in the skeletal muscle, and also in the steroid-producing cells, hormone-producing cells. They actually, they look pretty simple, but they are extremely complicated. Let's go now by one little other organelle that you thought I forgot, but I didn't forget, hopefully, and it is the mitochondrion. The mitochondria, there may be as many as 3,000 in one single cell, and are usually ovoid, and they have an external membrane that is smooth and an internal membrane that has many multiple folds, like this. Now, these mitochondria are obviously in the production of energy. They take up pyruvic acid or fatty acids, and they produce large amounts of ATP. One interesting fact about mitochondria is that those cells that have a large number of mitochondria, like the proximal convoluted tubule, for example, they tend to be acidophilic. In other words, they take up eosin more than hematoxylin. Of course, there are more interesting facts about the mitochondria. You probably remember something else, and that is that all our mitochondria are derived from our mothers. Why? Simply because at time of fertilization, the spermatozoon penetrates the ovum, including with the mitochondria. But what happens to the mitochondria? They die. They undergo autophagy, what they call it, and they are destroyed. So we only have maternal mitochondria, and that has given rise to a lot of things, to a lot of studies. For example, about 30, 40 years ago, there was a study made on ribosomal, pretty much on mitochondrial DNA, sorry, and which proved that all the humankind derived from a single woman about 100,000 years ago. That's one detail. But anyway, the mitochondria have circular DNA that allows them to multiply by themselves. In other words, they don't need to be produced by the cell. They themselves multiplied using their own circular type of DNA, which is to me amazing. All these organelles in general are motile, and they move around. The membrane is also perpetually rechanged, in perpetual change. Now, one detail. In the cytoplasm is not really just like a jello or a little simple gel. It is a universe of molecules. And it has a cytoskeleton, it has a supporting structure that gives it more strength, and also allows the cell to move. Some cells, like the neutrophils, the monocytes, they move like an amoeba, and especially whenever there is an infection, that's very important. So what kind of fibers we have in this called cytoskeleton? You have microfilaments, such as actin, which are very, very, very thin microfilaments, about 7 nanometers in thickness. We have other ones which are intermediate filaments, which are a little bit thicker, about 15 nanometers. And those are several types of keratin, vimentin, and others, like desmin, that can also be stained with immunoperoxidase. Okay, so we'll do the intermediate filaments, which is the one that we need to talk about. And that would be the microtubules. They look like a little tubule, but they are made of rounded units, alpha and beta units that polymerize and easily or rapidly form and disappear. And that is particularly important in one of the organelles, which is the centriole. I'll try to make it to you. The centriole is an organelle that's a T-shaped, and one of the arms of the centriole has 9 triplets of microtubules in one direction and 9 triplets in the other. There is also a centriole matrix, some other microtubules. And what do? They have 2 important functions. One is in the mitosis. The centriole divides in 2, multiplies, if you want to say, goes to both ends of the cell, and there is a dance of chromosomes in the center with microtubules between the centrioles. In addition to that, some of the cilia that have movement, some cells, the respiratory tree, respiratory mucosa, for example, they have centrioles like that are in charge of the whole situation. Now, like I told you, the cell is really a very complicated thing. How do we know that it is very complicated? Because it takes one cell to make what you are, what I am. All my cells are exactly a copy of the one that was made a few years ago. So it is, and now that's what we're beginning to understand with the field of molecular biology, which is very not very easy to pretty much to comprehend the magnitude of this universe. It's just extremely complex. It's my personal impression, which I cannot prove it to you, that probably one single cell is more complex than the entire universe that surrounds us the way we know it. I know it follows a lot of very complex laws, and it's very big, but the cell being very small is enormously, not only complex, but enormously important because it makes us. So now the final question, is there one cell in our normal body that works only for itself? I've asked myself that question. I was thinking about it, and I don't think there's one. All of them work in the setting of the other ones to make you what you are, to make me what I am. And that's not that's an important question. There's only, however, one cell that works only for itself. Only for itself, doesn't care about the other ones, and kills the organism, and that is the cancer cell. With this, I'll say goodbye in the meantime to you, and I'll be looking forward for the next class. Thank you.